All right, then, ready to dive in. Today, we're tackling a new study. And let me tell you, it has the potential to be huge EE for us equine vets.
Oh, yeah.
It's all about a new EHV-1 vaccine.
Okay, I'm intrigued.
This just came out last month, hot off the presses, from viruses.
Fresh research. Gotta love it.
So get this, we're talking about a live attenuated vaccine, and this team at a Xinjiang Agricultural University in China, they're targeting two very specific genes, ORF37 and ORF38.
Now, those are some interesting targets, especially with EHV-1.
Right, when I saw that, I was like, okay, now we're talking. These genes, they're mixed up in some of the worst parts of EHV-1, like how it can just hide out in the body.
Latency. Yep, tricky stuff.
And they used CRISPR-Cas9 technology.
Sounds futuristic.
It kind of is.
Yeah.
But it's real deal science. And they used it to snip out those two genes from the virus.
So they basically disarmed it.
In a way.
Yeah.
Yeah. Then they tested these, like, mutant strains in hamsters.
Hamsters, huh? So, like, the preliminary stuff.
Exactly.
Yeah.
But what's fascinating is they didn't just see if the vaccine stopped the hamsters from getting sick. They actually looked at how it worked in the body.
The mechanisms, that's the good stuff.
Right. Antibodies, T-cell activity, even how deleting those genes messes with the virus's ability to go latent. They were thorough.
Not all studies go that deep. That's promising.
Totally. So what was your first thought when you saw this paper? What jumped out at you?
Well, honestly, Figure 1, it's a visual representation of the whole mutant virus creation process. You can actually see what deleting ORF37 and ORF38 does.
Oh, interesting.
The cytopathic effects, the CPEs, they were way different between the regular virus and the mutant strains.
CPEs, now those are something our listeners might not be as familiar with. Can you paint us a picture of what those look like, under the microscope?
Okay, so picture healthy cells. They're plump, happy, just doing their thing. Now, introduce the regular EHV-1. It's like a demolition derby in there.
Yeah.
Cells shrinking, rounding up, detaching. It's brutal.
Yikes. Not good.
No, not at all. But with the double deletion mutant, the one missing both ORF37 and ORF38, still some changes, but it's more like a fender bender than a total wreck.
Okay, so less damage.
Yeah.
That makes sense. But did deleting those genes change how well the virus replicates? Because that seems pretty important if you're making a vaccine.
They definitely check that. Turns out the mutant viruses, especially that double deletion one, they did replicate a bit slower in the lab.
Slower? But did it actually affect the immune response?
That's the thing. It was subtle enough that it didn't seem to mess with the vaccine's ability to trigger a strong immune response.
So they found that sweet spot. Weak enough to be safe, but still strong enough to get the job done.
Exactly.
Okay, so we've got these mutant viruses. They look different under the microscope. They can still replicate.
Yeah.
What happened when they actually put it in the hamsters?
Well, first they wanted to compare, right? So they had hamsters infected with the regular EHV-1, some with the virus missing just ORF37, and others with the double deletion missing both genes.
Right, got to have a control group.
Exactly. And sadly, as you'd expect, the hamsters with the regular EHV-1, well, they didn't do well at all.
Yeah, that's the harsh reality of EHV-1.
Severe neurological signs, weight loss, and tragically, a lot of them died.
It's a tough reminder of why we do what we do,
Yeah.
Why we're always searching for better ways to fight this thing.
Absolutely. But here's the good news. The hamsters that got the double deletion mutant, the ones missing both ORF37 and ORF38, no real weight loss, no major clinical signs.
Don't leave me hanging.
None of them died.
Wow. Okay,
Yeah.
that's really significant.
It is. It really points to deleting both genes as being key for attenuating the virus, making it safer.
So what about the hamsters who got the virus missing only ORF38? How'd they do?
That's the nuance here, right? It's why they included that group. Those hamsters, they did have some weight loss, and a few showed mild or moderate symptoms, but importantly, no deaths in that group either.
So deleting both ORF37 and ORF38, that seems to be the winning ticket, at least in hamsters. But, and this is a big but.
We treat horses, not hamsters.
Exactly. So how do we know this will actually work in our patients?
Yeah, that's the million-dollar question right there, and that's why we can't jump the gun and get too excited just yet. You know, we got to be careful about taking what we see in hamsters and just assuming it'll be the same in horses.
Right, of course. Got to be realistic.
But even so, this research, it's a really good sign, a solid foundation to build on.
Okay, so what makes it so solid?
Well, they didn't stop at just looking at if the hamsters got sick or not. They wanted to get in there and really understand how this vaccine was working on a deeper level, so they started digging into the immune response.
Right, so let's unpack that a little, what they find.
They found that those hamsters who got the double deletion vaccine, the one without ORF37 and ORF38, they had way higher levels of these things called neutralizing antibodies compared to the other groups.
Okay, now I know we use the term antibodies a lot, but for those of us who might be thinking, wait a minute, what are those neutralizing antibodies again? Can you give us a little refresher? Like, why is this finding such a big deal?
Of course. So you've got your regular antibodies. They're like your first line of defense, right? They latch onto the virus and basically signal for the rest of the immune system to come and destroy it.
Yeah, like putting up a wanted poster. Hey, immune system, this guy right here.
Exactly. But neutralizing antibodies now, these are the big guns. They go a step further and they can actually block the virus from infecting cells in the first place.
Oh, wow. So they're like stopping it at the door before it can even cause any trouble.
That's it. And with EHV-1, which is notorious for spreading quickly, having those high levels of neutralizing antibodies could be a real game changer for us.
Yeah, that makes a lot of sense. So we've got the neutralizing antibodies on the scene, but what about the rest of the immune system's lineup? What about T cells? Where do they fit into all of this?
T cells, these are like the special ops team of your immune system.
Oh, I like this analogy. Go on.
They're the ones that come in and take out any cells that are already infected, which helps to stop the virus from spreading even further.
Okay, so they're like the cleanup crew, making sure the virus doesn't have a chance to settle in and multiply.
Exactly. And here's the best part. This study found that the hamsters who got that double deletion vaccine, they had a much stronger T cell response than the others.
Oh, wow. So this vaccine, it's like it's hitting the virus with everything it's got, neutralizing antibodies to block it at the start, and then a boost of T cells to take out any stragglers. Pretty impressive.
Right. It really seems to be working on multiple levels, and that's what you want. You want that multi-layered protection.
So we're talking about maybe even preventing those really awful EHM cases and potentially even reducing viral shedding, which would be a game changer when it comes to managing outbreaks.
It's definitely possible. But again, I got to remember those are hamsters, not horses.
Right. Always have to keep that in mind.
But it's exciting. This is good, solid research, and it gives us a really great place to start.
Okay, so speaking of starting, what are the next steps here? What else do we need to figure out before we can even think about actually using this vaccine in horses?
So first things first, we need to see these results hold up in actual horses. Hamsters are great and all, but their immune systems are different.
Right. We can't just go vaccinating all the horses based on a bunch of hamsters.
Exactly. We need bigger trials with horses, of course, to really confirm what we're seeing here and to figure out the best dose, how often we need to give it, things like that.
Okay. Makes sense. And what about long-term effects? Do we know if this protection sticks around? Will horses need boosters?
All good questions, and those need to be looked at too. How long does this protection last? Do horses need booster shots? And if so, how often? We need those answers.
Important stuff for sure. And how about safety? Do they have any bad reactions in the hamsters?
Another really important point, and in this study, they didn't see any adverse reactions in the hamsters, which is a good sign.
Oh, that's good to hear.
But again, we have to be really careful and watch for any side effects in the horses, especially once we start those bigger trials.
Of course, can't skip that step. Safety first.
Absolutely. And while this study didn't specifically look at viral shedding, that's another thing that we really need to dig into. Even if the vaccine stops horses from getting sick, we still need to know if they can spread it to others.
Right. We want to protect individual horses, but also stop those big outbreaks that can really devastate the industry.
Exactly. It's about the big picture.
Yeah.
And another thing, it'd be important to see if this vaccine works against all the different types of EHV-1. You know, viruses are tricky, they mutate.
So it needs to be effective no matter what it comes up against.
That's the goal. And lastly, don't forget the practical stuff, like actually giving the vaccine. This study used an intranasal method, so they put it right into the nose.
Which is different than how we usually vaccinate horses, right?
It is. Most equine vaccines are injections. So we have to figure out the logistics of it all. How easy is it to give? How do you store it? Is it going to be tough to do on young horses or ones that are difficult to handle?
Right. It's got to be something that works for vets and horse owners.
Exactly. But overall, I gotta say, this research is really exciting. It could really change how we deal with EHV-1, and that's a big deal.
It really is. It just shows you what we can do when we put our minds to it and how dedicated we are to keeping those horses healthy. And we're back. We've been digging into this really cool research on a potential new vaccine for EHV-1, and honestly, I'm feeling pretty optimistic. What about you?
Yeah, I am too. It's always good to see this kind of progress, you know? Gives us hope.
Totally. So before we wrap up, I want to circle back to one thing we touched on earlier. how this vaccine might affect viral shedding.
Ah, yes, that's the holy grail, isn't it?
Right. Because even if it prevents the disease, if we can also reduce shedding, that would be a total game changer, especially for managing outbreaks.
Absolutely. EHV-1, it's so contagious and it can spread through a whole herd before we even realize what's happening, even if a lot of the horses aren't showing symptoms yet.
Yeah, those silent shedders can be a real nightmare to deal with when you're trying to get an outbreak under control,
Exactly. They just keep spreading it without knowing it makes it so much harder to get ahead of it.
for sure. And that's why the idea of a vaccine that could reduce shedding, even if a horse does get infected, that's huge.
No doubt about it. It wouldn't just be about protecting individual horses anymore. It would be about protecting whole populations.
And that's what we want, right?
Yeah.
So this study, they didn't actually measure viral shedding directly, but they did find some pretty interesting clues in their data.
Oh, yeah, for sure. While they didn't measure shedding specifically, what they did find was that those hamsters that got the double deletion vaccine, the ones missing both genes, they had much lower amounts of virus in their systems overall, even after they were exposed to a really strong dose of EHV-1.
So even though they were up against a pretty nasty version of the virus, those vaccinated hamsters could still keep it under control, which suggests that they probably wouldn't be spreading as much of it either.
Right. So promising, but more research is needed, as always.
Of course, we got to confirm it in horses, but still, it's a really good sign. Imagine if we could significantly reduce how much EHV-1 is being silently shed.
That would be incredible for the horses, for the industry, for everyone.
It would change everything. Fewer outbreaks, fewer disruptions, just overall healthier horses. It's a great
Yeah.
goal to work toward.
It really is. And it just shows what we can accomplish when we put our minds to it, how dedicated we are to improving things for horses.
Couldn't agree more. Well, that's about all the time we have for today, but this is definitely a story to keep an eye on.
Oh, absolutely. I'm really excited to see where this research goes next.
Me too. Me too.
Yeah.
Well, for all you horse-loving science nerds out there, stay tuned because this is just the beginning. And from Vet Neuro Jar, keep those minds inspired, hearts light, and tails wagging.